Hello, I'm Dr. Sapna Thomas giving you a brief overview about the study we did at King's College London on the splicing factor SFPQ in motor neurons. Neurons are unique in that they are by far the longest cells in our body, receiving information from our senses and transforming it into behaviors. This role depends on intricate molecular mechanisms that we only start to understand. A substantial amount of the neuron's functions is done locally at synapses, located centimeters or even up to a meter away from the cell body and nucleus. During development, growth cones therefore need to take specific navigational decisions in a complex 3D space while being geographically very distant from the cell nucleus. Processes like axon navigation, axon and dendrite branching patterns, and local synapse formation all depend upon mechanisms of local decisions. We now know that these rely on mRNA transport and local translation that is being controlled by local signaling activities. In this study, we identified SFPQ as a splicing factor required outside of the nucleus for motor neuron differentiation. We find SFPQ in developing motor axons where it may be required for transport, to repress translation en route, to protect RNAs from degradation, for correct RNA localization, or to regulate local translation. We showed that at a stage when normal motor axons develop, cell-autonomous loss of SFPQ in motor neurons triggers loss of axonal transcripts and disables axon growth, preventing movement. These defects of neuronal transcripts, motor axon growth, and motor connectivity were rescued by a cytoplasmic version of SFPQ, demonstrating that the non-nuclear pool of splicing factor SFPQ is required for neuronal transcript maintenance, and it acts upon them to ensure normal axonal maturation. As SFPQ loss of function induces paralysis, we screened for mutations in the genome of human patients affected by motor degeneration. We found 2 mutations in 1 patient each that are not found in normal samples so far. These 2 mutations are present in a very special area of the protein, the coil-coil domain. Motor neurons expressing the mutated coil-coil domain SFPQ versions instead of the normal copy of the gene grow and connect to muscles, but they exhibit excess branching and have much less SFPQ proteins in axons. Thus, our study shows that the coil-coil domain is essential for normal axonal distribution of SFPQ, and it confirms the requirement of the axonal pool of the SFPQ for normal axonal morphology.